Replication is semi-discontinuous. Incorporation of radioactively labeled thymine nucleotides into DNA during replication, followed by autoradiography of the replicating DNA, reveals that the two strands of duplex DNA are both replicated at each advancing replication fork by DNA polymerase. DNA polymerase uses ssDNA as a template and makes a complementary strand by polymerizing deoxynucleotides in the order specified by their base pairing with bases in the template. DNA polymerases synthesize DNA only in a 5' to 3' direction, reading the anti-parallel template strand in a 3' to 5' sense. A dilemma arises. How does DNA polymerase copy the parent strand that runs in the 5' to 3' direction at the replication fork? It turns out that the replication is semi-discontinuous. As the DNA helix is unwound during its replication, the 3' to 5' strand can be copied continuously by DNA polymerase proceeding in the 5' to 3' direction behind the replication fork. The other parental strand is copied only when a sufficient stretch of its sequence has been exposed for DNA polymerase to move along in the 5' to 3' mode. Thus, one parental strand is copied continuously to give a newly synthesized copy, called the leading strand, at each replication fork. The other parental strand is copied in an intermittent or discontinuous mode to yield a set of fragments. These fragments are then joined to form an intact lagging strand. Overall, each of the two DNA duplexes produced in DNA replication contains one old and one new DNA strand, and half of the new strand was formed by leading strand synthesis, and the other half by lagging strand synthesis. And that is replication is semi-discontinuous. Have a great day.